All right. Thank you, Sal. And thanks everyone for being here. As Sal said, that I am the last speaker for today, for the course. And I think that my topics are both going to be a little different from what you've heard so far. First of all, the first hour, I'm going to talk about not X-rays. And so one thing that you may or may not know and probably heard in the very beginning of the presentations is that synchrotrons actually produce more than X-rays. They produce very broadband spectrum of light, including infrared. And so I am I was a beamline scientist at the old synchrotron NSLS-1 for 15 years, and I ran 2 infrared beamlines there, and we're just getting the infrared program up and running at NSLS-2. And what I'm going to talk about in the first hour today is some biological applications of the infrared microscopy. So there's a lot of infrared applications that you can do with the synchrotron, but my research has focused in biological applications, and that's what our beamlines were focused on. And so that's what I'm going to talk about today. And we're going to, and I'll explain the structure of the talk in just a minute, but the second hour then is going to be spent on talking about how to write a beam time proposal. So you've heard tons about how synchrotrons work and all the different techniques and applications that you can use synchrotrons for. And so we're going to close out the course by, I'll describe the how to write a beam time proposal, how to write a good beam time proposal with some tips and pitfalls. So it's a little bit different today than in the past. And I'll also say, back to this first hour, my presentation is what you want to hear. So I have, well, you probably can't see the number of slides, but I have 70 slides, and I'm not going to present them all. I'm going to have you tell me what you want to hear about, because I have 10 different applications. And so I'm going to introduce the technique first, and then I will, then I'm going to let the audience choose what topics you want to hear about, and we'll do that until Sal gives me the pulls the string at 10 o'clock, and we take our break. So all right, so with that, let me move on. If I can get the slides to change, maybe. Hello. Okay. So I would like to start with the acknowledgements. And so this is a photo of my research group, and this is a photo not from this past summer because nobody is wearing masks and we didn't have any on-site users from visitors from the summer, but this is 2 summers ago, the summer of 2019. And my research group actually has a wide range of group members. We have all the way from high school students to undergraduates to PhD students, and then scientists and engineers. And we all work together on both synchrotron X-ray and infrared applications of biological materials. And so I just want to give a shout out primarily to Tiffany here is now a postdoc in my group. She was a PhD student. She did her entire thesis with me at NSLS-2, and that's something that's an opportunity for anybody interested in doing thesis work. And also Ashwin Ambi, he is also currently a PhD student with me, and Adam Lowry. So the 3 of them are currently in the group doing research. I also have, in the summertime, Ruth Pietri is from the University of Puerto Rico. She comes with her undergraduate students, and Stephanie came that summer to do research with us. And we also have members of or participants from the DOE SULI program, so the Science Undergraduate Laboratory Internship. So if you know of undergrads interested in doing research at the synchrotron, that's also an opportunity. And finally, I'd like to super acknowledge Ryan and Randy, who are integral parts of the group. They are LightSource staff employees. Randy is an engineer, and Ryan is another beamline scientist, and he does a lot of X-ray fluorescence microscopy, which we do a lot of collaboration, because a lot of our samples that we do infrared on, we can also do X-ray studies. I'm not going to go into the X-rays today. You've heard a lot about the X-rays, but today I'm going to focus on the infrared. Okay, so I want to go, you've heard a lot about imaging in this course, and I just want to kind of reiterate when we think about imaging, what we actually mean. To me, and probably to the general public, if you think about imaging, it's really what we see, and that's what, and visible light is what our eyes see. So obviously from 400 to 700 nanometers in wavelength. But what we can also see is using photons from other wavelengths. So you've heard a lot about using X-rays to see chemical composition of materials using, for example, X-ray fluorescence microscopy. Visible light, so if we go up a little bit higher in energy, what a regular old light microscope will allow you to see is using visible light. But what I'm going to talk about today is using a lower energy wavelength of light, so we're seeing with infrared light. Most people are familiar with infrared imaging as it applies to thermal imaging, so things like night vision goggles, where it actually measures the heat given off of a sample, and so you get a heat map of your material. What I'm talking about with the infrared microscope today is a different wavelength range. It's a little bit longer, shorter wavelength infrared, and we actually do chemical imaging. And so I'm a chemist, I'm not a physicist, and maybe the only chemist to speak with you. I think Ira was also a chemist, but I'm going to speak to you today about from a chemistry perspective, because what we actually do is chemical imaging of biological materials. All right, so how does that actually work? So what we actually have is infrared spectroscopy or microspectroscopy is based on looking at the vibrational molecules in, or the vibrations in molecules. So if you were to take a look at the water molecule, for example, it's vibrating all the time. It's vibrating in an asymmetric way, or what we call anti-symmetric way, a symmetric way, or bending. And all of these vibrations are happening at different wavelengths. So the water OH stretching vibrations occur at 3277, around 3490, and then this bending mode is occurring around 1645. So if you were to put infrared light into water, and you're going to get absorptions at each one of these frequencies. And so this is an infrared absorption spectrum of water, where you can see these two OH stretches are right here creating this peak, and this OH bend is right here creating this peak. And so because of that, every bond in a molecule can actually be a fingerprint using infrared spectroscopy. So let me give you another example. If you take, for example, you look at a carbon-oxygen single bond versus a carbon-oxygen double bond. We all know that a carbon-oxygen double bond is shorter and tighter than a carbon-oxygen single bond. So that carbon-oxygen is going to vibrate faster or have a higher frequency than a carbon-oxygen single bond. So you can detect the difference between C single bond O, C double bond O with infrared spectroscopy. The same thing is true for if you change the atom size. And so a chlorine is smaller than a bromine. So this carbon-chlorine bond is going to vibrate faster than a carbon-bromine bond. And so you're going to get a different vibrational frequency in the infrared as well for those. So you can tell carbon-chlorine different from carbon-bromine using infrared spectroscopy. An example of an entire molecule with lots of different kinds of bonds is methyl methacrylate. So this is the infrared spectrum in red of methyl methacrylate. And what we can see is, first of all, there are a lot of CH stretches in this molecule. And so that's why we get an intense absorption features here of the CH stretch region. CH2s and CH3s vibrate a little bit differently from each other. So that's why there's multiple peaks under here. And then if we go down to lower frequency, the C double bond O bond here is a very intense peak right there. The C single bond O is a lower frequency, as I described up here. And then you also see things like the carbon-carbon single bond stretch. So if you didn't know that this was methyl methacrylate, you could actually take a spectrum of your unknown, and there are libraries out there of known compounds, and it would tell you that this is methyl methacrylate based on its bonds and its chemical structure and how it reflects in the infrared spectrum. All right, so one great thing about infrared spectroscopy is it is quantifiable. So it's an absorption technique where we basically measure the intensity of the infrared light before it goes through and after that goes through the sample. And by taking the negative log of that ratio, you get the absorption. And so this is what we call the Beer-Lambert Law, or Beer's Law for short, where the absorption is equal to the absorption coefficient times the thickness of the sample times the concentration. So proportionally, if the concentration of your sample goes up, the absorbance is going to go up. If the thickness of your sample goes up, the absorbance is going to go up. The absorption coefficient is it's a constant that is specific to the type of bond that you're talking about. So a C double bond O will have an absorption coefficient which is different than a C single bond O. So and basically that's what this is saying here is that the absorption coefficient for a C double bond O at 1740 where it absorbs is a large number, whereas at 1700 it's a smaller number. And so it's really just measuring what the propensity to absorb at that particular frequency will be. But that's a constant for bond type. All right, so the next thing I want to describe is the difference between infrared spectroscopy versus microspectroscopy. So you don't have to use a microscope to do infrared spectroscopy. Obviously, most is done on bulk samples. So if you have a powder and you want to know its infrared spectrum, the typical way of doing that, infrared light is very absorbing. So you'll take that sample, you grind it up in a mortar and pestle, you add potassium bromide or some other infrared transparent powder, you mix them together and you can press it into a pellet and take an infrared spectrum. So if you have a uniform bulk sample powder, you can do it that way. If you have a liquid, if you have a solution, you can do a similar type of thing. You can take your solution, you can sandwich it between two infrared transparent discs, and then you can close that together and you can take an infrared spectrum. So your typical infrared spectrometer has a beam in it anywhere from about 1 to 3 millimeters in size. So if your sample is uniform on that size scale, you don't need an infrared microscope and you don't need a synchrotron. However, if you have a sample that's very small or has microscopic heterogeneity, so this is your bulk sample, but you have these little pieces in there that are different from the rest, and you want to know what their infrared spectrum is, you want to focus your infrared beam down into a smaller spot, and then, or you can actually do a map of the entire sample, and I'll show you a bunch of maps today. You can do a map of the entire sample to get infrared spectra at every single pixel. The advantage here now, so you can, again, you can buy an infrared microscope that you can use in the lab. You don't need a synchrotron to do infrared microspectroscopy. There are great thermal sources out there called glow bars. But the spatial resolution is limited with the glow bar. So you can get down to 20 to 30 micron spatial resolution. However, if you want to improve that, traditionally, for traditional infrared microspectroscopy, you can get down to the diffraction limit, which is about 2 to 10 microns. Or you can do nano IR. That's a brand new technique out there where you can get all the way down to the nanoscale. Using an AFM IR type technique, which I can talk about a little bit later. So, but the synchrotron, because it's so much brighter than the glow bar source, will allow you to improve the spatial resolution. So if you have a sample that's heterogeneous on the 25, 30, or bigger scale, your conventional laboratory instrument is going to work fine. But if you want to go smaller than that, the synchrotron is going to help you out a lot. Okay, so this is how what we call spectroscopic microscopy works. You've heard about this for X-ray fluorescence microscopy. It's the same idea where you focus your beam down into a tiny spot, and then you move the sample stage and so that you can actually generate a map. So you can just focus point and shoot the beam at an individual spot that you want to look at. What you're seeing here is brain tissue. It's an autopsy brain tissue from a person that died from Alzheimer's disease. The bright green spots are what we call amyloid plaques in the brain, and those not supposed to be there. And that's what the hallmark of the disease. And we were interested in studying the structure of these amyloid beta plaques with the infrared microscope. So you can You can focus the beam just down onto these individual spots and get spectra of them, or you can actually raster scan your sample and get a spectrum at every single pixel, and then you can recreate an image later, and I'll show you a bunch of those in a little bit. All right. So since you don't have the opportunity to actually visit the synchrotron, I'll show you a picture of what an infrared microscope looks like. This is one of our 3 infrared microscopes that we have at Brookhaven. This is the newest of them. This is a Bruker Hyperion 3000 with a Vertex ADV spectrometer. So this spectrometer is where you would do spectroscopy. If you want to do small stuff, you put the beam into the microscope. The beam itself from the synchrotron comes down this pipe down here and into the spectrometer, and then into the infrared microscope from there. And so, and as you can see, at least to me, this looks very much like a microscope. So one of the big differences between X-ray microscopes and infrared microscopes is the visible light path to see your sample uses the same optics as the infrared light path to take data. So you don't have to register your, you don't have to make sure your camera is pointing at the right spot like you do with an X-ray microscope. The infrared light, red light, and the visible light all fall, onto the exact same spot, and then and you can take data. So they are very handy because you can learn how to use these in the lab, and so most of our users already know how to use an infrared microscope, and they just come to the synchrotron to get better spatial resolution for challenging samples. All right. So there are 2 types of ways, there are 2 types of infrared microscopes out there. There are microscopes where you can actually point and shoot onto the sample, or microscopes where you can actually get it using a pixel array detector. So the one I just showed you had a focal plane array detector on it, where you take the infrared light and you shine it down onto your sample and you recondense it. But in this particular case, you put a big beam down onto your sample and you actually use the detector pixels in order to do to resolve the image. So that's very different because you get many pixels at a time. The infrared microscope I just showed you will give you 128 by 128 pixels all at the same time. We don't fill that many. We probably fill about half of that, but it gives you the advantage of being able to take data faster, but you do get some pixel blurring, and so it's not quite as quick. This type of infrared camera is very much like just a digital camera that you have, except it senses, it can do infrared spectroscopy. So we use this if we have less challenging samples and we want to collect data faster. But if we have samples that we actually want the best spatial resolution, what we will do is use a single pixel detector here, and that allows us to just take one spectrum at a time, but you get the best spatial resolution. Okay, now every infrared microscope has 2 modes of data collection depending on what your sample is like. You can collect your data in transmission, or you can collect your data in a reflection mode. So the schematics you're looking at here are 2 different infrared. This is the infrared microscope objective, where the yellow is a reflective surface. So the infrared light will come through. These are Schwarzschild objectives, which work just like telescope objectives, except in reverse. The light will come through. It will reflect off of this mirrored surface, bounce off of this mirrored surface, and then focus down onto the sample. There's a matching objective on the underneath side of your sample that sends it to the detector if you're measuring in transmission. If you're measuring in reflection, what you have to actually do is block half of the input light so that you can get the output light back out again. So you send the light in, you reflect it and focus it down on your sample. It will reflect off the surface of your sample, bounce back up, and then there's what we call a pickoff mirror that will take the reflected signal and send it to the detector. Our preferred way of taking data is in transmission. So if you can make your sample thin enough and low enough concentration, we like to measure in transmission, but sometimes that's not possible. And then we will measure data in reflection. Okay, so since most of the applications I'm going to talk about today, actually all the applications that we'll talk about today are related to biological materials, I wanted to give you kind of a primer on what the infrared spectra look like for different biological materials. So if we look at the chemical structure of a protein here, a protein always has an amide backbone. There's a C double bond O and an NH, a carbon nitrogen in the backbone. So nitrogen, carbon, carbon, nitrogen, carbon, carbon is the amide backbone of the protein. Because of that, you always have what are called the amide 1 and amide 2 bands in a protein spectrum. Amide 1 is coming from this C double bond O. Amide 2 is coming from this carbon nitrogen bond. They're in every infrared spectrum, but they're different, as I'll show you in a second, depending on the structure of the protein. For lipids, or fats, oils are all lipids. They all have these long hydrocarbon chains on them. And because there's a lot of carbon-carbon bonds and a lot of carbon-hydrogen bonds, you get very intense CH stretching modes and reasonably intense carbon-carbon stretching modes. And so those are very specific to different types of lipids. And the way, whether you have branching or straight, for example, is going to give you different shape of your carbon-hydrogen stretching mode region there. In nucleic acids, so that's DNA, RNA, we have phosphate groups on them. And so it's the phosphate group that helps us to identify nucleic acids the best, because you have both an antisymmetric and a symmetric phosphate stretching mode down in the lower frequency region that doesn't overlap with the CH or the amide bonds. And carbohydrates, which are sugars, for example, and other types of carbs, all have this ring-like structure with these OH groups on them. So there's a lot of carbon to oxygen stretching modes, which are very intense. And then we have these OH stretching mode as well, which are a little harder to use because you always get this big, broad, fat peak. But the CO stretching mode is very different depending on whether you have a 5-membered ring, a 6-membered ring, whether there's an oxygen in the middle, whether there's some unsaturation, etc. So this is the fingerprint region for carbohydrates. So, but these are typical infrared spectra of biological materials. Now, if I talk just one more slide about proteins, because this is our favorite thing to look at. In my research, we do a lot of work on neurological protein folding diseases. So diseases that are caused by misfolded proteins in the body. Again, the protein backbone looks like this: carbon, carbon, nitrogen, carbon, carbon, nitrogen. So you always have this amide bond, this nitrogen, carbon, double bond, oxygen. And it gives rise to this amide 1 peak, which looks like a plain old peak. But if you actually look closely at it, if you stretch it out a little bit, it's not a simple Gaussian peak. It actually has lots of sub-peaks underneath it. And these all come from the secondary structure of proteins. So sometimes proteins are alpha helical, sometimes they form more of a sheet structure, sometimes they're very unordered. And each of these different environments around the carbonyl group is going to give rise to a peak that's slightly different in frequency. And so all these different frequencies, they kind of fall under this envelope, but when you take your data, if you have good quality data, you can actually deconvolve this amide 1 peak and find out how much helix unordered beta sheet turn you have in your sample. And that's really important for studying protein folding diseases, which you'll see a little bit later. All right. I want to just emphasize a little bit how the synchrotron is beneficial to doing infrared microspectroscopy, and I'd like to just do that with an example. So this light micrograph that you're seeing here are single red blood cells. They're some of the smallest cells in the human body. They're only between 5 and 10 microns in size. And if we want to study what's going on in these red blood cells, for example, malaria is something that affects red blood cells. And if we want to study malaria in an individual red blood cell, we have to focus our beam down to 5 or less microns in size. If we were to do that, if we were going to aperture and focus the beam down with the Globar, the laboratory-based source, this is what your spectrum is going to look like, this blue one. It's kind of noisy. But if you use the synchrotron, you get beautiful spectra all the way down to the diffraction limit. So we even get better than this today with the brightness of NSLS2. But you see the amide bands of the proteins in the single red blood cells are very well shown here in the synchrotron spectrum, but the globar spectrum, there's no way you would be able to deconvolve this amide-1 peak in order to find out things about secondary structure because of the noise in the globar spectrum. So if you want to do things that are really small, the synchrotron is helpful. It also helps with contrast if you use the full field infrared detector. So I described how you use the pixel array camera. This is just an example of spinal cord tissue looking at axonal projection. So these lines that you see here are regions of high lipid concentration from myelin in brain tissue. And what we were actually looking at was we wanted to be able to resolve these individual lines. And you can see if you take the data with a laboratory-based array detector, you get an image that looks like this, or an image that looks like this if you resample the data. But what we do with the synchrotron is we have so many pixels that we can actually resample, we can actually oversample, I should say, using a 0.5 micron pixel. So we're not getting 0.5 micron pixel resolution because of the diffraction limit. But if you oversample, if you take multiple spectra at each pixel, sorry, if you take a spectrum with a pixel size like this, you minimizing the overlap so that you can get a much more detailed image, even though you're not you're not beating the diffraction limit, but you're helping to make a sharper image. And you can just see that in example spectra here, where you can see how noisy it is using the globar source, but how nice it is using the synchrotron source, all the way down to the 0.5 micron pixel resolution. So, so that's another way of looking at primarily tissues. If you want to look at larger areas, but you want to get a very high spatial resolution, you can do that with the synchrotron. All right, so one more slide about sample preparation, and then we're going to go on to the examples. So this is it for biological materials. We typically see things that are either in a tissue form or in a cell form. And if you have a tissue, the way we typically prepare samples is by taking it and embedding it in some sort of embedding material. And then we cut it into thin sections. And so it's basically just using we use a microtome, which is not anything fancier than like a meat slicer in the deli. We cut those samples very thin, we place them on an infrared transparent slide, and then you can take data from them. You can't get infrared light through an entire block of sample. It's not like X-rays that will penetrate. You have to have 10 micron or so thin samples. We also look at biological cells. They're easier because they're only about 10 microns thick anyway. We can grow those cells in culture and we can spin them onto an infrared slide, or we can actually grow those cells right on an infrared transparent slide and take data from them. So either way allows us to do imaging of cells. Okay, so that is basically my, my brief introduction and summary of how infrared microspectroscopy works. The rest of the talk, so the next half hour, we're going to spend on applications. I have, as I mentioned in the beginning, I have a bunch of them. There's actually 10 of them, and I don't have time in 30 minutes to talk about all 10. They're, some are a little longer than others, some are a little shorter, but I'm going to let the audience now vote for what topics you would like to hear about. And I will just talk on these different topics for the next half hour. We'll see how many we get through. So I'm going to have Dennis, I think, is going to launch our poll if he's still there. So he's going to launch a poll, and we're going to have everybody check off 2 topics that you would like to hear about, and we're going to go in the favorite order in terms of which ones I'm going to present. So everybody go ahead and check off the 2 that you'd like to hear about the most, and then we'll see how the poll comes out. How do we do, Dennis?
We're about 3/4 of people voted, looks like, almost, and we're at the 2 to 3 minute mark.
I think that's fine.
Okay. I'm going to end the poll in like another 10 seconds, folks, so lock in your votes.
Okay.
All right,
I don't know if you can see it or not. I know the attendees will now be able to view the poll results.
Yeah.
I
So
see it.
You see it?
Sally, you got it captured? I'm not sure it's gonna. I think I have to close it in order to change my
I have
slide.
it. So the top one is cancer.
Okay, so I see that I'm going to do biopolymers first, and then we'll do cancer, only because I think biopolymers has some useful stuff, and they almost are a tie. And then and then we can go from there based on what you got. So all right, okay.
Stop sharing now, or I can leave it open, and everyone can just close their poll window. It should be okay if they want, or move to the side.
Yeah, I think they can close, and as long as Sal or you can tell me what to do third, because I know the first two. So
I got it.
all right, so we're going to do biopolymers and biocatalysis first. And with every good synchrotron experiment, there's a question that our users, we ask when users are coming, what question they're trying to answer using the infrared microscope. And so with the biocatalysis problem, this project was all about improving the efficiency of immobilized enzymes on polymers. And so just as a bit of background, there is a whole field out there of doing biocatalysis, or just catalysis in general, using enzymes, because we know that enzymes are very efficient catalysts. They display a high regioenantioselectivity selectivity, and they're less hazardous in polluting. But the problem is with enzymes is that they require water to work. So you can't throw an enzyme into toluene and expect it to actually do its job, because it's just going to denature and fall apart. So one of the answers to that is to actually use something called immobilized enzymes. And one of them out there on the market right now is called Novozyme 435, which is commonly used. It's an immobilized lipase. So this is an enzyme that is immobilized on a polymethyl methacrylate bead. And so what you can do is you can throw this bead with the enzyme, into, for example, organic solvents, and you can get it to do its job. But the efficiency of this process depends on how much enzyme you need to make the bead and the distribution of the enzyme in the bead. So the company and the researcher that came to us was very interested in improving this process in terms of the protein concentration and distribution. So this is what we had to work with. This is a cross-section of a Novozyme bead. This is the bead, and it was embedded in wax, which is what you're seeing around it, and the bead is right here in the center. The infrared spectrum of methyl methacrylate looks like this blue one right here. So the bead all by itself has a spectrum that looks like this. If you take a spectrum of the bead with the protein in it, it looks like the red one. And so if we subtract those two, the pink one is the infrared spectrum of the protein, the lipase itself. And that makes sense, because here's our amide 1 and here's our amide 2. The other thing we want to make sure is that our embedding compound, which was paraffin wax at this time, doesn't interfere with the spectrum. We also, at another point in the study, which I won't talk about today, but we also were interested in studying the product of the lipase reaction, and that was polystyrene. And so we needed to know what its spectrum looked like too. But the bottom line here is what they wanted to know was the distribution of the protein within this bead. And so you can't obviously see it from the visible light image, but you can actually take a infrared image of it. You take a spectrum at every pixel, and then what you do is you integrate this peak, the amide 1 peak, which is the protein, and what we found is this ring around the outside. And so the bottom line of this story here was that the enzyme itself was not doing any, it wasn't getting into the center of the bead, so it couldn't act well to improve, to run the reaction. And so they had all this wasted space in the center of the bead, and it was only working on the surface. And so what they did is they went back a little bit to the drawing board, and they shrunk the size of the bead so that they would actually build a bead that the enzyme was doing its job all the way through the center of the bead. And so that was the result of that, was that we were very surprised to see that the enzyme was only on the surface of the bead in this 80 micron shell and not in the center. Now I have another example, and that has to do with polymer degradation. And in this particular case, the question we were asking was all about the photo-oxidation products of a polymer exposed to sunlight. So there's a lot of, plastics, polymers that are exposed to sunlight, and you don't want them to break down over time. And so the question was, what happens when you expose polymers to sunlight over time? And in this particular example, it was polystyrene. So this is what polystyrene looks like. These are 5 micron size polystyrene beads. And what we wanted to do is actually watch over the course of time what was happening. So this was a time-resolved experiment where we put these polystyrene beads under the infrared microscope. We shine UV light on them, and we watch over the course of 14 hours what's happening to the infrared spectrum. And what you can see here is that this particular peak, this is your CH stretching mode, your regular aliphatic CH stretch is disappearing over time, and you're getting this aromatic CH stretch growing, which is showing oxidation of the polystyrene, and essentially it's breaking it down. And we could actually see that with the growth of, here's the CO, and then there's the disappearance of the and the appearance of the appearance of C double bond O, which is the oxidation, and appearance of the and disappearance of the aromatic CH, which is here. So we can actually take those data and generate a movie of what happened. So this was a full field infrared. So we were looking at all 4 beads all at the same time with the infrared microscope. And we can watch the appearance of the carbonyl as the oxidation happens, and we can watch the disappearance of the CH aromatic as the oxidation happens. And this is jumping around only because of the registration from, because we were doing this over 14 hours of time. The stage didn't sit exactly still, but you can see you go completely from almost no carbonyl to a lot of carbonyl after 14 hours, and lots of CH aromatic to almost no CH aromatic over time. And so that's just an example of how we can do the infrared imaging to look at organic polymers or mixtures of polymers in systems. Okay, so that's the biopolymer example. I'm going to stop there and see if anybody has any questions, because it's easier for me to ask questions at the end of each particular topic than to ask questions, at the very end. So we can also, you can post questions during the break too. But if anybody has any questions now, you can put them in the QA, I think, right, Sal? And then you can.
Yes, please use the QA if you have any questions.
And I'll just give you 10 or 15 seconds if anybody has any.
No questions so far.
Okay. All right, well, if there aren't more questions, I'm going to move on to the cancer example, but you can put them into the QA anytime and we can answer them after the break as well. So cancer is it was all about tumor growth, this particular example. And in this particular case, we were interested in understanding something called matrix metalloproteinases and how they degrade healthy tissue. So what do I mean by a matrix metalloproteinase? Well, basically MMPs for short are enzymes that a tumor has that will basically degrade or chew away the healthy tissue around them. So as a tumor grows, it needs to have space to grow. And in order for this tumor to make space, it has to degrade or remove some of this the healthy tissue around it. And so the question, and it's MMPs, these matrix metalloproteinase, that do that. And so the question that we had was whether these MMP enzymes, do they actually get secreted by the tumor, or are they on the surface of the tumor and not secreted? If they're secreted, that means that they could work a lot further away from the tumor in order to break down the healthy tissue. But if they're just on the surface, then they can only act very close to the edge of the tumor. So we did this in a model compound study. This is work done with Irit Sagi from the Weizmann Institute. And we use a model tissue. So their healthy tissue, you can use a model compound called gelatin, and that's the simplest model of tissue. We made a gelatin film on an infrared transparent material. And then we grew cells, we grew tumor cells, HT1080 tumor cells on top of them. And we did infrared imaging again over the course of time. I'm not going to show you a movie in this particular case, but just as a static snapshot of this is the infrared image here down below. Now, what we're looking for is we're not looking at the cell itself, we're looking at the composition of the gelatin. Because if the gelatin is undegraded, it has an infrared spectrum that looks like the green. And if it's degraded, it has an infrared spectrum that looks like the red. You get a shift in the peak by about 30 wave numbers when the gelatin breaks down. And so what we did here in the infrared image is we plotted 1625, which is the degraded gelatin divided by the undegraded gelatin. And so where you see the high, the red, that's showing you where the gelatin was degraded. This is the cell in the middle. You don't need to pay attention to that because it's going to look different than the gelatin itself. But what I want you to look at is the red versus the yellow versus the green out here. And you can see that the high gelatin degradation actually extends almost 15 to 20 microns away from the edge of the cell. And so this image is really telling us that the cell was able to secrete the MMP enzyme into the matrix so that it can do more damage in terms of breaking down the cell, the matrix around it, instead of being very close up. So this is a short study. I mean, a short example, but this was the cancer example that I have. So does anybody have any questions on this one?
There is one question from Armando. How can we improve the resolution pixel issue?
How can what kind of resolution do you want? Are we talking nanoscale?
Yes.
Yeah. Okay. Of course. So the infrared spectroscopy itself is limited by the wavelength of light. And so diffraction limit allows you to just get to, for mid-infrared spectroscopy, gets you to about 2 to 10 microns. However, we are now developing, well, I'm not saying we're developing, is being developed AFM IR, where you can actually use it, you can shine your infrared light onto and actually my Alzheimer's example has an example of this, but you can shine your infrared light onto the tip of the AFM probe, and then basically all you're measuring is when the AFM probe tip hits the surface of the sample, you're measuring just that volume of sample. So you can do nano IR like that. And that's just growing. It's just starting to grow at synchrotrons now, and just starting to be developed, because you need a nice bright source of infrared to shine onto the AFM tip in order to do that. So the way it actually works is you would do an AFM image of your sample, and then you could actually do AFM IR on particular regions that you are interested in. So that's right nowadays, that's the going go-to technique for improving down to, and you can get down to, depending on whether you're using thermal detection versus scattering detection, you can get down anywhere from say 20 or 30 nanometers to 100 nanometers using nano IR. Okay, more questions, or should I move on to another one? guess that answered Armando. Okay, Sal, what do you want to do next?
All right, the next one is bioremediation.
All righty. Where are we? Okay. All right. So this is an example of bioremediation of toxins from the soil. And so this is a study to see whether bacteria can actually remove toxins from contaminated soils. So in particular, this was a study looking at dredging. And for those of you that aren't familiar with dredging, what that is you, it's if you have a river or a lake or a channel that boats need to get through, and the water isn't deep enough because soil accumulates in it, they will come in and they will dredge soil away, and dredge soil out with a big machine, and they will take it away. Now, that's all fine and dandy as long as there's no contaminants in the soil that you're moving. But if you have contaminants in that soil, what can happen is if they're tightly bound to the soil, then you're going to be okay. But if they're not tightly bound to the soil, when you disrupt this soil, you're going to release the contaminants into the water, and it's going to kill the fish and the plants and all the other wildlife around. And so they were very interested, this group, Upal, in understanding whether how different contaminants bind to different particles in the soil, and whether it's dangerous to the being released in the water. So the contaminants they were looking at were called polycyclic aromatic hydrocarbons. These are exactly like that. They have lots of rings in them, and they're aromatic rings in them that are hydrocarbons. And they are very toxic to plants and fish. And so their question was, they wanted to look at silica particles, which are sand, versus black particles in the soil, which is organic matter. And they wanted to see which of these. So they were taking individual grains that had mixtures of both. So there was some bare silica, and then there were these darker areas here that actually had organic matter on them. And because these particles are very small and heterogeneous, we could take the infrared microscope and focus down just on the bare silica, and just focus, and then also focus down onto the darker area. And we actually did an infrared map of the organic matter of the polycyclic aromatic hydrocarbon peaks in this particular area here. And what you could see is that this area, the bare silica, had no contamination, whereas the organic matter did. So it was to no surprise, but it made it very clear that the organic matter attached to these silica particles were absorbing the PAHs. And the next step, which I don't have an example of, but the next step here was then, with this known information, they were actually interested in treating these samples with bacteria, healthy bacteria, not those that make us sick, in order to basically eat away the polycyclic aromatic hydrocarbon when they go and eat the organic matter. There's another example which I want to put into my presentation for in the future, where this is done in a very bulk method, where there was an oil spill, and they used bacteria to different group, but they actually used bacteria to remediate the oil from the surface of surface of the water, but then also surface of rocks and everything else inside the water, in order to break down these, in this case, not PAHs, but break down oil. So this was just a particular example of how they can actually use bacteria to break down these PAHs once they know where they're actually located. Okay. Any questions on this one? All right. What do we want to hear next, Sal?
All right, the next one is plants and biofuels.
Plants and biofuels. Okay. Okay, so this is actually the PhD work of my PhD student, Tiffany Victor, who I showed her picture in the very beginning. And we're actually, in this particular case, looking at plant cell walls and trying to understand how they affect biodegradability. So if you want to use a plant as a biofuel, what you're interested in using is the cell walls of these plants. So this is a cross section of a poplar, and it's the cell wall that you want to use. However, before it can be a biofuel, you actually have to break it down. And that's what we call recalcitrance. So if you want to make a plant less recalcitrant or more able to convert the cellulose from the plant cell wall into ethanol, which is what biofuels do, you have to be able to reduce the plant recalcitrance. And so this picture here is just an infrared, a set of infrared images from the poplar cross section. This is just our cartoon, but this is the cross section of all the different layers in the poplar. This is a typical infrared spectrum of a plant. There are tons of peaks, and they can really all be assigned to different things. But what I want to point out most importantly here is what we call the 1740 peak, which is an acyl group, which I'll get to on the next slide, and also lignin. The bigger this peak is, the harder it is to break down a plant. But all of these infrared images were taken from the exact same map, but just by integrating different peaks, you can learn about the lignin, the polysaccharides, the proteins, etc. So our collaborator, CJ Liu at Brookhaven Lab, was very interested. He's a geneticist, he's a biologist, and he's interested in growing genetically modifying plants so that to reduce their recalcitrance, to make them better biofuels. And this is his wild type plant. This is tobacco, and it's a wild type tobacco plant. He genetically modified it. And what he actually did was he genetically modified the acyl ester group. And so what he wanted to do is if you reduce the amount of acyl groups, you can acyl esterification is something that makes cells more recalcitrant. So if you reduce this acyl and the acyl esterification, you're going to improve the ability for it to break down. So he wanted to know if his genetic mutation actually worked if you reduce the acyl groups, and that's what we did with the infrared microscope here. This is just a visible light image of the wild type and the transgenic. Here's your cell wall, and this is the infrared image of the acyl group. So in the wild type, you can see it's red. There's very high acyl group. In the transgenic, it's green. These are plotted on the same scale, and it's green, and so you have a much lower acyl group. The infrared individual spectra kind of show the same thing, that you're reducing the acyl group in the transgenic versus in the wild type. And this reduction was confirming to him that his acyl esterification was going down and improving the ability improving the biofuel capability for the tobacco. The only downside of the whole project is that so that was wonderful. He was super excited, but on the actual biotechnology side, if you take a look at this plant, it's nice and green. It's actually healthy. But because you're reducing the acyl esterification, it doesn't grow very big or very tall. It falls over and it wrinkles up. And so there's still this balance between trying to improve making it a better biofuel, but at the same time allowing it to grow like a normal plant does. So, okay, so that was that one. Any questions on that one? No? Should we do one more?
Yeah, let's do one more. So the next one would be bone disease.
Bone disease. All right. All right. So bone disease, and in particular, the disease that we look at is osteoporosis and treatments for osteoporosis. So bone is a terrific sample, or I shouldn't say bone is a terrific sample, but infrared is a terrific technique for studying bone. Because bone actually has both protein and mineral components. So your collagen is the primary protein component in bone, and it's what makes bones flexible. And then hydroxyapatite is the primary mineral component in bone, and that's what makes them stiff so that you can stand up. And what happens in osteoporosis is this is what normal bone might look like, normal trabecular bone. This is in osteoporosis, you lose bone. So you actually lose, your body dissolves away part of the bone, which is, first of all, a bad thing. But the question we had was whether the composition of the bone that's left behind is affected. And so this study was actually done in well, it was a study done where monkeys were treated with steroids in order to see whether they would improve the bone formation in osteoporosis. And so in a monkey, monkeys don't get osteoporosis naturally. So, but if you overactinize them, if you take their ovaries out, they will develop osteoporosis. And what you're looking at here, this is the knee joint of monkey. And incidentally, these studies were done not for this particular infrared project, but the studies were done for drug treatment about 5 years before we actually did the infrared study. So this is my qualifier that no monkeys were harmed or sacrificed for this particular study. It was extra information that we learned from this work. But this is the knee joint of a monkey. This is the cartilage up here. The darker area is the bone right underneath it. And what happens during this study is at the time the monkey is overactimized, they give them a fluorescent label that goes to the site of new bone. And so right at the time of overrectomy, they gave them this green label. And so we can find the bone using a light microscope that was only 1 year old right after the 2 years old, right after the overrectomy. And then we can look at this orange area that was given 1 year after the overrectomy. And what we can do with the infrared microscope is focus down onto these individual areas. So we don't want to know the composition of all the bone, because this is old bone and new bone mixed together. We want to know just about the new bone, and we want to know whether osteoporosis is affecting it. So what we found here, these are infrared spectra. Red goes with the red 3 pixels, green goes with the green 3 pixels. And what we really wanted to know, this is your mineral peak, this is your phosphate peak. So if you look at the green ones, you can see that it's very big. And so that means that the bone produced right after overrectomy was still healthy bone. But if you look 2 years after overrectomy, you can see that these peaks are a lot shorter. It's about half the size of what they're supposed to be. And so what that's telling us is that bone composition is actually affected with osteoporosis, and more specifically that the bone mineral formation is slowed down. So not only is your body losing old existing bone, when it's making new bone, which it does until death. So you replace 15% of your skeleton every year, but the quality of that new bone that's being produced is actually lower than normal in osteoporosis. And without the infrared microscope, we wouldn't be able to see that, because you would be averaging everything out over the whole skeleton. But using these labels in the infrared microscope, we can see things in more detail. So another study we did with bone had to do with microcracks, and also something that they've also discovered in osteoporosis is that one of the common treatments for osteoporosis are drugs called bisphosphonates, and they do a great job at slowing bone loss. But the question was, with these bisphosphonates, whether the chemistry of the bone, the composition of the bone, was actually affected in the process of bisphosphonate treatment. And it was shown just using light microscopy that something called microdamage, or these microcracks, were increasing in bisphosphonate treated animals. And so this is just a photograph here. Again, it's a light microscope with a stained tissue. So this pink area is where you have a microcrack in the bone. They're very small. So they're, maybe they're 100 microns long, but just a few microns wide. It's not something you would feel. These are not like hairline fractures or something. They're very, very small. But if they accumulate, then it can make bone break. So what we wanted to do is look at the chemical composition of the bone around the microcrack. And that's what these images are all right here. And so we were looking at the carbonate content, the phosphate, the acid phosphate, crystallinity, and collagen cross-linking. And what we did find then is that the bone composition is different around the microcrack. So the crack is forming, but you get changes in the carbonate, you get changes in the acid phosphate, and you get dramatic changes in the collagen around this bone. So as microcracks accumulate, the composition of your bone is progressively getting different, and this accumulation could definitely reduce the increase the propensity for microfractures. So Okay, that's the end of the bone story, and I'm looking at the clock and thinking it's probably time to take a break, Sal? What do you think?
Yeah, let's take a 10-minute break. We'll come back at 10:15 New York time, or 15 minutes past the hour, your local time. So,
And if anybody has questions, you can put them in the chat and we can address those first. Otherwise, the next topic will be completely different. It'll be all about writing beam time proposals.
so there is one question
Okay.
for the bone samples.
Okay.
Want to answer that now, or should we answer that later?
We can do it now while we're thinking about it.
All right. How are the bone samples prepared?
The ones that I showed you, those bone samples are all embedded in methyl methacrylate. So there's a couple ways of preparing bone samples, but the ones that I showed you, they were embedded in methyl methacrylate, and we used a microtome to cut them 5 microns thick. What we're doing more of now is actually not doing the embedding. We actually can polish the bone just with a regular polisher, and we can actually take data in reflection mode instead of in transmission mode. And the users like that a whole lot better because it's really hard to cut thin sections of bone. So you can actually just polish it and measure in reflection mode and get the same kind of information. So that's the more popular way of doing it now.
All right, another bone-related question. Have you studied teeth, the effect of fluorination from toothpaste or other agents?
So that is a great question. We have not done any teeth studies, but it would be really an interesting one to do. I've always, that's not my group's field of research, but I've always waited for, for a user to write a proposal on fluorinated bone, but we haven't done it yet because it could be very interesting to see how, fluorinated bone. In all these studies, you have to have a control and you have to have it treated. That's, my number one message for doing biological, samples is, but if we had treated versus untreated fluorinated bone, it would be a great study to do, but we haven't done it.
All right, there's 2 more general questions. You want to answer them now, or should we wait?
Yeah, sure, as long as they're coming now.
So how does the IR differ from Raman spectroscopy?
That's another great question. So infrared spectroscopy is an absorption technique. So it's really simply the intensity of the light going in and the intensity of the light going out and measuring how much gets absorbed. Raman spectroscopy is a scattering technique. So actually you take your infrared light and you scatter it off the sample. And then if the sample absorbs it, you get less scattering back at those particular wavelengths. And so Raman spectroscopy is scattering as compared to absorption. The disadvantage to Raman is that because it's a scattering technique, the efficiency is something like 10 to the minus 3, 10 to the minus 4, where absorption you get much better data easier. The advantage to Raman spectroscopy is that you can actually, the probe size, you can use very, very small laser beams to do the scattering. And so you don't have the diffraction limit issue with Raman scattering as much as you do with infrared. So it used to be that Raman microscopy was kind of the way of doing higher resolution infrared, but the signal to noise just wasn't very good. And so now that's kind of transitioned over from microscopy to doing the AFM IR instead.
All right, and there is one more. Can you use IR on fossil carbon like coal?
Not easily, because first of all, it's very absorbing, and secondly, the carbon or carbon black or graphite is kind of just all the same kind of carbon. So the spectra are very bland. It's not like it's an organic material, so it's a tougher thing to do. If you were looking for coal amongst something else, it would be very nice study, but it would be hard to study just coal unless you were looking for organics or something else in it. But coal itself is a tough one. Yeah.
All right, and I think that's the last of it for now.
All right.
So let's take that 10-minute break. We'll come back at 10:20. Is that all right?
Okay. Sounds good to me.
All right, so we'll come back at 10:20 or 20 minutes past the hour, your local time. Thanks. Hi, Lisa. There's two questions on the Q&A.
I see.
One-
Should I answer them now or wait till, it's the same I
It's
think
the
it's
same
a
question.
question and a clarification.
Uh,
Should I answer them now or are we waiting still?
let's start. It's already 10:20.
Okay. All right. So the question is about identifying protein isomers, for example, beta, gamma, actin. Unfortunately, infrared is not sensitive enough to identify isomers unless you were doing it in like solution, or if it was just single form of the protein. So you couldn't do it in tissue, or you couldn't do it in cells or something, but you could do it if you had a protein. If you had a protein solution, for example, and you wanted to know whether it was whether you had one of the two isoforms. But the difference between the two isoforms is so small that you wouldn't be able to distinguish that difference if it was mixed with other proteins, for example. I hope that answers her question. All right, should we move on to the other? Did anybody have any last questions about the infrared part? Because we're completely changing topics.
I guess not. So let's move on.
All righty, let me share this guy. You seeing it?
Not yet.
Through. Oh, hold on. I need to hit the share button first. Okay.
There we go. Research facility, logistics, and how to write a proposal.
Yeah, that's. Let's try to get it in presentation mode.
And it's in presentation mode.
Awesome. Okay. All right. So on for something completely different. This is kind of the finale to the course. You've spent the last, I don't know how many weeks, learning all about synchrotrons and the value of synchrotrons for research. And so what I want to do in the next probably half hour or a little bit more is to describe how user facilities actually work and how to write a good beam time proposal. So I understand that everybody had a little bit of homework from all of the lectures. And so I'm going to tell you what your homework is first to help you pay attention while we're going through these slides. So the homework for this part of the talk, I don't have any homework for the infrared part, but this is the homework for the for this part of the talk, is to write the abstract, which has to be 250 words or less, for a beam time proposal based on your thesis research. So if you don't have thesis research, you can write an abstract on anything you would like, you think the synchrotron would be useful. And it doesn't have to use infrared microspectroscopy. You could use any of the X-ray techniques that you used, for example, or it could even be developing something new to build a synchrotron. It could be accelerator based, etc. But take a stab at writing an abstract for a beam time proposal. Okay. All right. So let me. You've heard this probably from Toon at the very beginning, but I'll just kind of bring it all back together again and remind you or let you know that, you know, NSLS2 at Brookhaven is a national user facility. And basically that means that they're open to anyone worldwide to come and use them. The Department of Energy actually has 27 national user facilities in the United States, and tens of thousands of scientists use them every year to do experiments. And so they're not just synchrotrons. These user facilities can be supercomputers, particle accelerators, the light sources, of course, neutron sources, nanocenters. There's a genomic center, etc. And so if you have a need for techniques that you don't necessarily have in your laboratory or access to, you can find a lot of these and the expertise to help you do those experiments at the synchrotron. And the greatest thing about synchrotron access is that as long as it's non-proprietary research, it's peer-reviewed access, which is free. And that's about 90% of the users that come to use the facilities. In the United States, there are 4 synchrotrons. There's one in Berkeley, there's one in Stanford, there's one near Chicago in Argonne, and then there's Brookhaven, which is on Long Island. And so they all operate the same way, in the sense that you have to write a beam time proposal. And if that proposal is good enough, it will get allocated beam time. And with that comes the help of a beamline scientist like me to help you do your experiments and collect your data. If it's proprietary work, which probably doesn't apply to most of you, we do charge a fee, but it's just full cost recovery, so it's not very expensive. And then there's other mechanisms to access the synchrotrons, like strategic partnerships and collaborative agreements, and that happens with industry, with individual universities, companies, etc. So. But what I'm focusing on today is this first part. It's the peer-reviewed access and how to be successful at getting access to synchrotrons. So at NSLS1 and then NSLS2, we have upwards of, well, not this past year because of COVID, but we have more than 2,000 researchers have used NSLS2. And we also have a nano center at Brookhaven. They have upwards of 500 users each year, and they come from all over the world. They come from academia, industry, other national labs, and they're all different types, all faculty, postdocs, grad students, undergrad, high school students, teachers, industry, etc. And it's very varied how much time they stay. They may stay for a few hours or days. Some are longer term resident users. And as you can see, we have students that actually do their whole thesis while they're at Brookhaven. And because of that, because of the diversity of our users, we have a diversity of different, beamlines and instruments, which you've heard a lot about already. So the actual proposal process kind of looks like this, and it works the same for NSLS2, the synchrotron, and also the nano center. And so the Center for Functional Nanomaterials is a CFN. That's a nano center. They also do it the same way that with the proposal process. And very soon you'll be able to write a single proposal to access both facilities together, but right now you have to write 2. The facilities typically operate on the order of 5,000 hours per year, and we have 3 proposal cycles per year. So there's the January to April, where the proposal deadline is the prior September 30th. May through August, the proposal deadline is the prior January 31. And September and December, the deadline is May 31st. So our next deadline coming up is January 31st, and that is for the May through August beam time cycle, May through August 2021 beam time cycle. We get, between the NSLS2 and CFN, we get upwards of 1,000 proposals and time requests every single cycle. And about a third to a half of them actually get instrument time. The lifetime of a proposal is variable, so it can be anywhere between 1 to 9 cycles, depending on the proposal type. Our typical proposal is what's called a general user proposal, which I'll get to on my next slide. Those are good for a year. However, because we have 3 cycles in that given year, if you want more time each cycle, you have to put in a time request each cycle that the instrument is requested. And so it's not like you would submit a proposal and then you get time throughout the course of the year. Every cycle, at each deadline, you would have to ask for more time on the proposal. And that's something that we try to remind everybody, but you don't want to forget, because if you forget to put in a time request, you can't get more time. You can request multiple instruments or beamlines on a single proposal. However, right now you can't cross over between the nano center and the synchrotron, but you can ask for multiple beamlines on one proposal at NSLS2. And that's why I mentioned soon we'll be able to submit them both. The best place to go to learn all of this information that I'm talking about, you'll have these slides, but this is also the user guides for each of the 2 facilities is the best way to find this information on the web. All right, so let me describe really quickly the different types of proposals that we have. But the one that applies primarily to everyone listening is probably this general user proposal. And I mentioned at NSLS2, these are good for 1 year. At the 1 year in the Nano Center, they're good for 2. And this is the most common form of user access for routinely supported techniques. And so if it's a single PI proposal, looking for routine techniques, then that's the general user proposal. And that accounts for upwards of, I would say, more than 80% of the proposals we get are general user. There's another form of proposal that is like the general user, but it's called rapid access. So if you have some sort of if you need rapid access to the beamliner instrument for hot topics or for straightforward experiments that have a fast turnaround time, so you just need a few hours for your final piece of data to publish a paper or write your thesis, you can submit a rapid access proposal for that. They're only good for 1 cycle, and you only get a small amount of time. And so they're very specific for this type of thing. And they're graded that way too by the review panel. So if you put something in as rapid access because you forgot to submit a general user proposal, they won't let you, they won't allocate it. We also have something called partner user proposals. These are groups who want to enhance the facility capabilities or contribute to the facility operation. Typically it's with staff or instrumentation. They basically get a fraction of the beam time in exchange for what they're contributing to the facility. So we have a lot of partner users on our beamlines who get somewhere typically between around 10 to 15% of the beam time in order for their group to run. And in exchange, they will basically put for example, put a person as a beamline staff on the beamline. They may contribute a vacuum chamber or an end station, a microscope, something. They may actually we have some groups that built whole beamlines, and then they end up getting 50% of the beam time. But those are our partner users. And we have about 20 of them right now at NSLS2. We also have a proposal called a block allocation group. This is something only at NSLS2, and they're good for 2 years. This is for groups of researchers that want to combine a short beam time request into a single proposal to permit greater flexibility. So the primary use of the what we call BAG proposals or block allocation proposals is our protein crystallographers. They come together as a team, perhaps from a single university or a single medical school where they might have 5 or 6 crystallographers, but everybody only needs a little bit of time and nobody knows exactly when their crystals are going to be ready. So they write this joint BAG proposal that basically gives them a small amount of shifts each cycle, but they're kind of guaranteed that time. And then they decide which one of their researchers on the BAG proposal get the time each cycle. So we don't micromanage these bags. They get to choose who's going to get the time. And then lastly, we do accept proprietary proposals. Again, that's full cost recovery instrument time. And those are typically about 3 years in length, but they often get renewed and get extended because we have a number of companies that have regular access to beam time, primarily, again, in the protein crystallography field for doing beam time. So. All right, so those are the different proposal types, but what I'm talking about, today is focusing on the general user proposal, but I'm happy to describe the others more if you want to learn more. Now, let me describe how the proposal flow actually works. So if you're out there and you're writing a proposal, the first thing that obviously happens is you submit that proposal. And I'm going to tell you all the elements of what gets submitted in a second. But the first thing that will happen is it goes for a feasibility review. So that means it goes to the beamline scientists and they read it and they make sure that it can actually be done on their beamline, because it may be something that they don't have the capabilities for. You might pick the wrong beamline, then we send it to another beamline. So it gets a feasibility review first. If it fails the feasibility review, that's the end of your proposal. So the message here is to make sure that you talk to the beamline scientist before submitting that proposal to make sure it's not going to fail feasibility review, because it's a lot of work to put a proposal together and then have it get declined right away. Once it passes feasibility review, it goes on to scientific peer review. So we have 6 proposal review panels, all specializing in different technique areas and scientific areas. and the proposal gets assigned to a proposal review panel, and it gets reviewed and scored, and then the best scored proposal are the one that get time. So they all get scores. An allocation decision gets made based on whether it's a good score or not a good score. If it's not a good score, you're not going to get beam time, obviously. If it has a good score above the cutoff, you're going to get allocated beam time. So you're going to get this wonderful email that says, congratulations, you've been allocated beam time on beamline XYZ, and you'll be contacted by the beamline scientist for scheduling. So that's the next thing that happens, is that we send out this congratulations, you got beam time, and then you'll hear from the beamline scientist who schedules your beam time. And right away, you have to do your guest registration and your safety approval form, because all proposals, once they get allocated time, you have to have a safety review. And this has to be done within no later than 2 weeks prior to the beam time. And if that happens, then you actually have your beam time. So once you have your beam time, just a reminder that if you want more time on that proposal, so there's still 2 more cycles left. If you want to ask for more time, you have to put in a new request. It doesn't get reviewed again. It just gets into the allocation cycle. If you have run out of time, either all the time that they gave you is used up, or your 1 year is up, then your proposal will expire and you have to submit a new one. So that's actually how the proposal process works. And this is true for basically it's true for most synchrotrons around the world, because there are upwards of 40 of them, maybe more depending on how you count. But definitely all the synchrotrons in the United States have this exact same proposal flow. Some have 2 and not 3 beam time cycles, but otherwise it's all the same. Okay, so the next thing I want to do is describe a little bit about how to write a good proposal, because you've learned all about how the synchrotron works, which means you're interested in synchrotrons. And so in order to use the synchrotron, you have to write a good proposal. So these are just some helpful tips in order to do that. So we're going to have a little fun with this one. And if you have any questions, type them in the Q&A and we can address them at the end. So step 1, the first thing you want to do is visit the websites, the user guides for either the Nanocenter or NSLS2, and the links are right there. You need to kind of decide what beamline you want to use, and make sure that that beamline is going to help you, is going to answer the problem that you want to solve. So once you visit the user guides, you have to review the following information. You have to decide what proposal type you want. It's probably going to be general user proposal. Then you can download a Word template where you can actually write that proposal. You can't upload the Word file to the proposal system. You have to copy and paste it, but this template helps you to share it with collaborators and other members of your group, for example, in order to work on it back and forth. And then once you're happy with it, you can put it into the online system. You want to review the review criteria and make sure your proposal are addressing the review criteria. Make sure you know the proposal deadlines, because if you miss it, you got to wait for 4 months for the next one. And then also familiarize yourself with things like user appointments, trainings, and other logistics. So everyone that gets allocated beam time has to register as a user, and they have to do some training. We are doing a lot of remote access beam time right now, obviously because of COVID. And even users who are just logging in remotely all have to have user appointments. And so as soon as that allocation happens, we encourage folks to go and get their user appointment. There is some training. It's computer training, cybersecurity. And if you are coming on site, there's other logistics they have to know about, because we do have on-site housing, dormitories and apartments if you want to stay, right on site during beam time. Okay, so that was step 1, is to do your reading and familiarize yourself with the process. The next step is to contact the beamline scientist or the instrument scientist. We have very friendly people at every single beamline who are going to help you to write that proposal. And so if you have no experience doing this and you've never done this the first time, it will definitely help you to contact the beamline scientist. And it's very common to collaborate on new projects, to collaborate with the beamline scientist on your project. You may do that for the first, proposal or first set of experiments, and then once you learn what you're doing, you don't need their help as collaborator anymore, and you move on to doing things on your own. But in the beginning, it's really helpful to collaborate with the beamline scientist. These are the types of questions that you want to discuss with them. You want to make sure that the beamline meets the specifications for your project. You want to make sure that they have the widgets and gadgets that you may need. So do you need additional instrumentation for a non-standard configuration? If you're going to do a vacuum chamber experiment, for example, you want to make sure that that they let you put your kind of sample into the vacuum chamber. If you need gases, you want to make sure that we have access to those gases and that our safety procedures allow them. If you have instrumentation you're bringing yourself, we have to make sure that that's all approved. And if they have instrumentation at the beamline that they will be supplying for you, you have to discuss that. And a really important thing to do is also discuss how much time it's going to take to do your experiment. So it's really hard, if I talked for an hour on infrared microscopy and I showed you a lot of infrared microscope images, but never once, I don't think, did I say, well, this one took 5 minutes and this one took 4 days. And so it's really sometimes hard to gauge when you're first starting to do this, how much time it's going to take to do one spectrum, one map, one image. And you need to put that into your proposal in order to justify the time that you're asking for. And lastly, this is a really important one too. If you have a sample that is kind of non-standard, you can ask the beamline scientist to do a feasibility check for you. So you can actually send them the sample. And we can't do this for all users, but if you ask, it's always possible to just test the sample, because you don't want to spend hours and hours and hours and days and months preparing samples the way you think they're going to work, and then have it not work when you come for beam time. And so it's always good to make sure that you discuss the sample prep and maybe even test the sample before you actually come for your real time. All right. After that, you've done your reading from the website, you've done your talking to the beamline scientist, and now it's time to sit down and write. And the first thing you have to do is actually write your title and the abstract. And I manage the proposal review process at NSLS2, and so I know all of our reviewers, and I know that abstract is the most important thing that you have to write, because if the problem is not clearly explained in that title and the abstract, it's hard to get further. This is also true for scientific papers. So proposal is like a mini scientific paper. So the title and abstract are, one point I have to make about this one is that we do have to report the title of your the title of your proposal and the abstract to the Department of Energy. So not that anybody is doing any proprietary, I'm not describing proprietary research here, but that's something to be aware of, that the contents of your proposal are completely private except for the title and the abstract. So, but it only goes to DOE. We're not providing those to anybody else. We're not putting them online, nothing like that. It gets sent in a secure document once a year to DOE for them to have the record of. All right, so that's your title and that's your abstract, and it's your abstract that is your homework. So the next steps in the proposal process is to describe the scientific importance of your proposal. So the reviewers that are reviewing your proposal are not experts like you are in the scientific area. They have a general, if it's condensed matter physics, they may have a general knowledge of many areas, but only know specific types of superconductors. And you have to keep that in mind when you're writing your proposal. So you can't get into the nitty-gritty detail with lots of acronyms and things, because they're not going to understand it. So you have to explain it at the level of a scientist, non-specialist, about what's known about the sample. That's the first thing you say. And then, why is it important? And then what is the hypothesis that you're going to test? So why do you need the synchrotron to study it? So I'll give you an example. Here's an example of a sample project that we have. We're studying world hunger, and we want to improve plant growth in poor soils. So this is what we want, you know, the importance of it is studying world hunger, and how we're going to address world hunger is to improve plant growth in poor soils. So that's a terrific, what is the, you know, what is the importance and what's the science, you know, the specific science you want to do. Now that doesn't say anything about how the synchrotron is going to help you. So that's where the hypothesis has to come in next, and what you're going to do with the synchrotron to test it. So here is an example applied to this one. We want to use X-ray spectroscopy at NSLS2 to measure the titanium pre-edge peak. And we hypothesize that the titanium pre-edge is indicative of tetrahedral titanium, which helps to retain phosphorus in soils, and this helps bring nutrients into plants. So it's bringing all the way back, you know, improving plant growth in poor soils. We know that plants need phosphorus. Phosphorus will help bring nutrients, including phosphorus, into the plants. And we know, and what we're studying is titanium, and how the titanium structure is important to retaining phosphorus in soils. And so it's bringing exactly what you want to do. We're going to do X-ray absorption spectroscopy, and we're going to study titanium, and we're going to look at the pre-edge. We're going to test whether it's tetrahedral, and that's how it's going to help to study those plants. So you want to bring it all together in the hypothesis, and that should really be in the clearly described briefly in the abstract, but also in the introduction of the proposal. The next thing you have to do is your research plan. So this is the bigger section of the proposal, where you have to provide literature background and correlative measurements or other lab work that support your hypothesis. So back to our titanium example again in world hunger, it's really helpful to the reviewers to know that you've done your homework as far as prepping the sample, and that you know what you have. So we've made measurements in the lab with artificial mixtures of soils, and added varying amounts of titanium dioxide, and treated them with phosphorus fertilizers. And we found that the soil pH is critical between 6 and 10. So these are all experiments that have already been done in the lab to make sure to narrow down the experimental range of samples that you want to look at. Then you have to also explain specifically what you want to do in a stepwise fashion. So this is what you've already done at home in the lab to support your hypothesis. Then you want to say, okay, in our experiments at the beamline, we're going to measure titanium XFs of 12 samples with the pH between 6 and 10, with varying amounts of titanium and phosphorus. You're going to put how much, titanium and how much phosphorus in there. Then you're going to say, okay, if this happens, we're going to do this next. If that happens, this is what we're going to do this next. So you have to really describe what you want to do and what the outcomes might be in order to justify the time. In addition, in the research plan, you have to justify that time specifically. So in the previous one, you said you're going to run so many samples with a pH of such and such and titanium phosphorus concentration of such and such. But then there has to be a time calculation in there. So we're going to say, okay, we've got 10 samples with 6 conditions, so that's a total of 60 samples. We know by talking to the beamline scientists that it takes 10 minutes to collect one spectrum and 10 minutes to change samples. So therefore, we need 1,200 minutes or 20 hours to collect the data on those 60 samples. So we also need, after talking to the beamline scientist, about 4 hours for setup time, which is about 1 day of time for these experiments. So this is the level of detail the reviewers love to see, because it really shows that you've thought about these experiments, what you're going to do in a stepwise fashion, and how much time it's going to take for every step. Okay, and then you also want to describe how you're going to analyze the data. So don't assume the reviewers know what you're going to do with the data once you get it, because, okay, you just collected 60 XF spectra, but what are you going to do next? So you're going to tell them, you're going to say, okay, we're going to use FEF, and we're going to simulate 4 versus 6 coordinate titanium on the computer ahead of time and make sure that you can tell the difference between the two. Then with the simulated spectra, we will compare those to the actually collected spectra So we're confident that we can actually use XFs to distinguish the difference between titanium 4 and 6 coordinate. So again, you have to describe how you want to analyze the data too. All right. Then we have a section, every synchrotron has a section where you have to describe yourself and who you are. And basically to justify that you know what you're doing on this proposal. So if you're a first-time user, we really like to see that you're going to collaborate with the beamline scientists, because it's just very helpful when you're first starting to do synchrotron experiments. Are you a student or a postdoc? And if the answer to that is yes, you should not be the principal investigator on that proposal. Your PI should be. And then but you should tell us about him or her too. So tell us about tell us that you're a graduate student or a postdoc. Tell us if you've had any experience in this particular area. And also tell us what the background is for your principal investigator. So maybe you've never done a synchrotron experiment before, but your PI has been doing synchrotron experiments forever. So make sure you say that. And then describe whether you're a regular or experienced user, because that really helps provide the confidence. If you're not collaborating with the beamline staff, it helps to provide the confidence to the reviewer that you'll be able to execute the work. And ways of demonstrating that you're an experienced user is by publications first and foremost in the scientific area of the proposal, so that we know that you're qualified to do the work. One thing that the reviewers don't like to see is a CV with 100 publications on it. None of them have anything to do with the synchrotron or the project that you're doing. We really want to focus on your expertise in doing synchrotron experiments in your particular research area. Okay, so we're gonna talk about yourself and your collaborators on the proposal. And then you are gonna have to create a safety approval form. This doesn't happen before you submit the proposal, even though I've got it in here as seeming that it does. The safety approval form actually comes after the allocation of the proposal, but it has to describe all of the hazards associated with your experiment, and it has to be submitted within no later than 14 days prior to the start of beam time. If it comes in closer to the beam time than that, it's possible that you'll have your beam time, that you won't get your beam time allocated and somebody else will get it. So especially if they're complicated safety related experiments, it's important to put that safety approval form in well ahead of time. Okay, so that's the meat of your proposal. Once you've done all of that, the last step here is to really allow the beamline scientist or the instrument scientist to take a look at it. So you downloaded the Word template and you wrote your proposal. You had all of your research group collaborators at home look at it. Everybody's happy with it. Then it's really helpful to send it back to the beamline scientists that you talked to in the beginning, and you say, do you see anything that I need to fix? And they will take a look at it and give you feedback. The most important step here, though, is to not do it in like the last week before a proposal cycle deadline, because they're probably getting dozens of these to review. And you can't expect them to respond to a lot of last minute requests, because they're doing all kinds of other things too, supporting users at the beamline. So plan ahead. If January 31st is the next proposal deadline, write that proposal in December and send it to the beamline scientists and have them take a look at it. Give them plenty of time to review it so that you can get it submitted well ahead of the proposal deadline. All right. Now, I just want to describe the reviewer evaluation criteria, because this is actually what the reviewers are in particular looking at when they're reading your proposal. Number one, they're looking at the scientific or technological merit of the proposal. So you need to get the point across, what the problem is you're trying to study and why it's important. The next step is to, considering the specifications and the facilities of NSLS2 or the Nano Center, is the work good use of the facility and expertise? So if you're going to write a proposal to do infrared spectroscopy on some uniform powder, that's not going to review well because we don't need a synchrotron to do that. We can do that with our lab-based infrared spectrometer. So it's important to justify the need for using NSLS2. What are the characteristics of the beamline or the ring that are important? It's important to describe the principal investigator and everybody else on the proposal, their track record for previous work. So we don't dock anybody for not having done, we want new users. And so we're not saying, oh, they've never done a synchrotron experiment before, we're not going to give them beam time. That's quite to the contrary. 1/3 of our users every year are new users. And so we really want new users, but we also want to make sure that they have written solid proposals so that they can get that data and then write paper. And then proposal well written. Is it clear what the proposers want to do, and have the scientific ideas been well thought out? We very often see beam time proposals that are just copies and pastes from grant proposals, and that really doesn't, it's a good place to start, but copying and pasting and submitting is not going to include the information that we really need to hear, like justification of the beam time and and that type of thing. So it is important to make sure the proposal is well written. So in terms of scoring here, I don't have the numbers on this slide, but the most heavily weighted section is the scientific or technological merit, and then these other three are about equally weighted in the proposal. All right, so I will just close with some useful guidelines and then some pitfalls, and I'll take any questions if you have them. But the first thing is some useful guidelines when you're talking about the scientific merit. So that's the most important criteria that gets reviewed on. Make sure you explain why this is important, and explain it again to the scientist, non-specialist, and not to a researcher that is into the nitty-gritty details of what you're doing. Number two, but equally important, is that hypothesis. I know that most of the reviewers are looking for the actual phrase that says, we hypothesize that. Because if you don't see that there, it doesn't mean, in some way we need to know what you're testing and how the synchrotron is going to test it. The likelihood of success for the experiment are dependent on the team's experience and track record, or collaboration with the beamline staff. Reporting any homework that's already been done so far. So did you do lab XRD, TEM, NMR, etc.? Do you have supporting data that, again, say why you want to use the synchrotron to study these samples? Make sure the samples, you prove that the samples are well characterized. So even if you're taking dredged soil, from the bottom of the Hudson River, they can still be characterized ahead of time. The particle size can be characterized. The composition of the water that they came from could be characterized. They could be separated by particle size, etc. So they're, even for the dirtiest samples, you can characterize. You need to characterize those ahead of time. And there needs to be a clear work plan. So sample numbers, identification of them, what are the environment instrument parameters, how much time does it take per sample? So really do the math here and describe how much time it's going to take you to do these experiments. And lastly, something we often see missing is the clear outcome. So, with these results, we will be able to show X. So it's really the synchrotron experiments are gonna show X, and so that's that's important to see too. All right, and I know this is a funny cartoon, but this is just it's so very true when it comes to our reviewers who have to review a real wide breadth of different scientific areas. We only fund those proposals we can understand. Just make it understandable. Don't make it so complicated that nobody knows what you're talking about. All right, and then that kind of just leads into some pitfalls, things that we see often in reviewing proposals that aren't so great. First of all is samples that are unidentified or poorly described. It's very common for people to say that they're gonna, study those the soil samples with titanium dioxide, but not say what the concentration of titanium is, or not say how much phosphorus fertilizer is in there. They're just going to say generic things about their samples. They won't talk about the setup requirements. So, do you need the beamline to, tune the energy all the way up to 30 keV, which is going to take them an extra half a day? Do you need them to focus the beam down to a half a micron instead of 2 microns, which might take them an extra half a day? Those setup requirements are important. The workflow and work plan needs to be well described again. And make sure you talk about data processing and analysis. So don't assume that just once you get the data, the reviewer is going to know what you're going to do with it. You have to say what you're going to do with it to get the answer that to test the hypothesis. And it's very common that folks don't justify the instrument time. So, doing that math to say we have this many samples, each one's going to take this much time, and so we estimate that we need this much beam time. And even if you if you have to double that in the end because it's you're not sure it's going to take this long, that's still fine. At least it shows that you thought about it. And then things that are unclear. So it's important that once you get the data, this is what you're going to learn from it. And generic statements we see frequently. This is one. This is one that is straight from a proposal. The measured data will be analyzed to obtain valuable information. That sentence could or could not be in the proposal, and it would have no bearing on the proposal whatsoever. It's completely generic and doesn't add anything to that proposal. So, it's really the XF's data will be analyzed to obtain the titanium 4 versus 6 coordination state, and that will tell us that phosphorus is it retains phosphorus better with the titanium 4 and making it a better plants grow better because it retains phosphorus. You have to get into the details. And then also placing the experiments in context with other methods and modeling. So those are just, some of the common pitfalls that we see in proposals that don't get beam time, and we don't want that to happen to you. We would like everybody who spent all this time taking this class learning about synchrotron techniques to write proposals and actually come to NSLS2, whether it be remotely or in person for beam time. And so the last step here is to really to do a great job on the proposal. So with that, I am going to close and leave you with one more funny cartoon that I tend to like, and ask if there's any questions. And but that's all I have for today.
All right, there is one question right now. How can you discuss the setup requirements if you are a first timer?
That comes from talking to the beamline scientist again. So I think that if you tell them what your samples are all about, what you want to study, they will help you with describing how the sample preparation should work and what instrument setup will be necessary. So it really comes down to speaking with the beamline staff to get that information for the proposal.
All right, I have another question. If you have proposal time and then you're requesting more time, do you get that time within the same period, or do you need to wait for the next period?
You have to wait for the next period. So the way it works, and I didn't go into exact detail, but I'll give you an example. If you write a proposal and you say you want 12 shifts, for the lifetime, for the 1-year lifetime of the proposal, the PRP, the proposal review panel, will review it, and they'll say, okay, this is a great proposal. We're going to give it a score of 1.5, which is an excellent score. The best score is 1 and the worst score is 5. We're going to give it a score of 1.5, and we agree that they get 12 shifts, but in the first cycle, we're only going to give them 3 shifts. So that means that in your first cycle, in the first 4 months, you'll get 3 shifts, but then at the next proposal deadline, you have to ask for 3 more or 6 more, and then you'll get more. So you can't get multiple you only get the amount you're allocated for in each shift. Now, if they said we're going to give you 12 shifts for the lifetime, and you get allocated 12 shifts in the first cycle, and you use those up in the first cycle, then your proposal expires. So you actually have to write a whole new proposal, even though a year didn't go by. So there's two types of scenarios there that you can actually come across that are, it's important. But you'll get an email if you use up all your shifts and the proposal expires before a year. But to answer your specific question, yeah, if you you have to ask for more time on that proposal in the next cycle. You won't get it more than one allocation in one cycle.
Are there any restrictions regarding nationalities because of COVID for pre-essential experiments?
Does that mean coming on site? Is that what you mean? So right now, because of COVID, we are only accepting users that are coming from states contiguous to New York. So we're not international users or even users from California. Our international is a no for sure. Users from California, they have to quarantine for 2 weeks before they're actually allowed, not at Brookhaven, somewhere else in New York before they're allowed to come. So for this current cycle, September through December, and the January to April cycle, I know that we're only having very few users come on site, and they're almost, actually all of them so far have come from either New York or states that touch New York. But I'm hoping if things look, things are looking promising, we're hoping that by next summer, we'll be able to open up to users from everywhere that the travel policies allow. But we don't have any restrictions as far as foreign nationals. In normal times, there are no restrictions as far as foreign nationals doing experiments at any of the synchrotrons. So that's all fine. But because of COVID travel restrictions, we've had to limit it at NSLS too. But you can do it remotely still. So it doesn't mean, coming on site is great, but if experiments can be done by shipping the sample to the beamline scientists and collaborating with them to get it done. A lot of people are doing that now.
All right. If there's any more questions, please write them in the Q&A. Right now, there are no more questions. All right.
I see half the question. If the experiments require different beamlines, what's the second half of the question? Crashella. Yes. Okay. So yes, if the proposal requires more than one beamline, you should talk to each of the beamline scientists just to make sure. Typically, a proposal there'll be a primary beamline, and then there will be others, that you would also need or like to use. But it's never a bad idea to talk to each of them. And when you do, tell them that you talk to the other one too, so that everybody's on the same page. We're a small place, and all the beamline scientists, know and talk to each other, every day. And so it's not like there isn't communication, but it's best to come from the user if you want to use multiple beamlines for your project. You're welcome. Anything else?
All right. If there are no more questions, I think we could call it a day.
All right.
All right. Last minute. All right. Guess not. So, oh, there we go.
Yes. So she asked whether proposals can include participants from different countries, and the answer is absolutely yes. There aren't any restrictions again on you know, who experimenters are or where users come from, and so it's not a problem. That's not a problem at all. And we do encourage everyone that's participating, even remotely or preparing samples, you know, they would all be part of the proposal. And so it's perfectly fine to have, and it's expected that, you know, all collaborators would be on the proposal, even if, regardless of, you know, where they're coming from. And if you're collaborating with the Beamline staff, they should also be on the proposal too, because that gives confidence to the reviewers that you've spoken with them and that they're on board with helping with the experiment.
All right, so if there are no more questions, I guess we'll just call it a day. Once again, I would like to thank everybody for participating in the NSLS 2 lecture series. And hopefully we'll be able to see you around if we get past COVID. Yeah.
No, when?
When we get past COVID.
Yes, exactly. Start thinking about those proposals now so that we're ready when everybody's got the vaccine and we can all see each other again.
All righty.
Thanks, everyone.